Awesome. Thank you so much for that introduction, Araceli. So just like Araceli said, today we will be playing around with the CLC main workbench. We'll be doing some Sanger sequencing alignments. We'll be doing some cloning, primer design, and some in silico experimentation. So of course, first things first, we have our legal disclaimer. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. Now, we love to keep these webinars as interactive as possible. So I am going to ask a couple favors from you guys. So the first one is going to be to fill out some polls that will be popping up sporadically throughout the meeting. It's a great way for us to kind of keep track of things and collect feedback from you guys to make sure that we are doing our best job possible for these webinars. Secondly, if you have any questions that arise during the presentation, please utilize the Q&A box. We have some folks hanging out behind the scenes today that will be taking questions from that Q&A box and answering them live. I will also take a couple breaks throughout the webinar today and grab questions from that Q&A box to answer live as well. And just like Araceli said, we will email you a copy of the presentation for today. So, please be on the lookout for that, as well as you're able to also see the recording from today's presentation by visiting that same registration link that you had clicked to sign up today. And then lastly, at the bottom of the screen, you'll notice that there is a raise hand button. So this is a place where I will be asking you guys questions throughout the webinar just to get some quick feedback. So we'll be utilizing that raise hands button. And that, of course, is right next door to our Q&A box here. So let's just make sure that everything is working appropriately for today. So if you guys could go ahead and click on that raise hand button, that would be really helpful. Excellent. Looks like a couple of you. Yeah, awesome. A few of you have definitely found that raise hand button. So thank you so much for double checking that with me. So here's a quick agenda for what we'll be going over today. Of course, like I said, we'll be playing in the CLC main workbench here. So I'll give you guys a brief introduction as to what the main workbench is and what it isn't. And then we'll quickly jump right into the software itself, where we will be performing some alignment and then a phylogenetic tree from that alignment to have a look at the lineage of some of the samples that we'll be importing here. And then we will quickly move on to Sanger sequencing analysis. So I'll show you guys how to import, perform a standalone trimming, as well as an assembly. And then we'll go through and I'll show you how to resolve any conflicts so we end up with a really high quality consensus sequence. And then our last outing for today is going to be a cloning experiment. So I'll show you guys how to create primers and set up for restriction-based cloning, as well as perform an in silico cloning experiment itself. So with that, here's some of the different visualizations that we will be going through today. So like I said, first thing on deck is going to be creating an alignment of multiple different sequences here. You can see we have listed out here a whole bunch of different sequences, and we are aligning all of them to try to figure out where the differences between all of these sequences arise. And then, of course, we're going to take that alignment and we are going to use that to generate a phylogenetic tree from that alignment here. And then once we've created this tree, we'll actually be able to overlay in some metadata information to have look at not only the lineage of all of these different bugs or sequences from these bugs that we have collected in our samples, but maybe have a look at identifying metadata characteristics. Like for instance, these seem to be definitely sorting by what type of water they were collected in. So I'll show you guys how to do that. And again, color and manipulate that phylogenetic tree based on some metadata here. And then next we'll launch into some Sanger sequencing analysis. So I'll show you how to import some of your traces from your Sanger sequencing experiments. We'll create an assembly from those traces, line them to a reference genome, and then, of course, we'll go through and have a look at some of the conflicts that might be arising between our traces and our reference genome shown here at the top. So I'll show you how to resolve these conflicts and how to make sure that your consensus sequence is of the utmost quality that it could possibly be. And then lastly, we'll be generating some visualizations using the molecular biology tools. So here we're going to go ahead and use or create a construct using a vector and an insert sequence. And while we're doing that, we'll be designing some primers. We will be trying to figure out what types of restriction enzyme sites we want to use and tack on to the end of our primers. And then finally, we'll perform an in silico cloning experiment to make sure that everything is checking out before we even spend time at the bench performing these experiments. So I am going to launch a quick poll right now just to check in with you guys to make sure that these are indeed some of the visualizations that you would like to see generated today. And if there are some visualizations that I didn't cover, feel free to add those into the Q&A box or into the chat box. And if we have some time at the end or perhaps during our Q&A breaks, we can cover those as well. So awesome. Looks like you guys are helping us out by participating in those polls. So thank you so, so much. Now let's get into the meat and potatoes of today's presentation. So first things first, what is the QIAGEN CLC main workbench? Well, what it is a really user-friendly desktop genomics application. So this is going to allow us to do all sorts of different secondary analysis and numerous molecular biology techniques without having to have any sort of programming or command line experience, or even any sort of bioinformatics experience. Now, this is widely used across a whole bunch of different facets of biology, whether that's in academia, out in government, in industry. And there's lots of different tools that are in the CLC main workbench that you guys can use. And I've created these little tiles on the side of this slide here, just to give you an idea of all the different things that we can do here. So of course, today we're going to be performing some cloning and primer design, looking at some Sanger sequencing, but maybe you have some microarrays that you'd like to do some gene expression analysis on. There are tools that are included in the CLC main workbench. Now, one thing to mention is you might notice that there is something missing from this list, perhaps. So one thing that the CLC main workbench does not have tools for is going to be any NGS-based experiments here. So if you are generating NGS data alongside your Sanger sequencing data, you might want to consider upgrading to a CLC Genomics Workbench, and that will unleash all sorts of other tools for your NGS based data, as well as things like single cell sequencing and so on and so forth. Right, so now that you have an overview of what the CLC main workbench is, let's get right into it. So first things first, we are going to create an alignment from some sequences and generate a phylogenetic tree to have a look at the lineage of some of those sequences from the bugs that we have collected from those samples here. So what I'm going to do is I'm actually going to pop right over into the CLC main workbench interface and get things started. So let's just take a brief tour around the application itself just to figure out where everything is and show you guys some of the most popular features here. So of course, at the top of the screen, we're going to have a whole bunch of quick reference buttons here. So import, if you're importing any sort of sequence information, sequence data, we'll be using that one a lot today. If you do have some sort of tools that you want to just quickly launch, we have this little cute rocket ship icon that you can go and launch some of your favorite tools. And then moving down, we have our navigation area. So this is where everything that you will be creating in the CLC environment will live. So you can think of this as like a Windows Explorer folders, just to keep things organized so you can quickly come back and sort through all of the different data that you've generated here. Now below that, we have our toolbox. So this toolbox is going to contain a wealth of information. So the first one that I do want to show you guys is this processes tab. When we click on that tab, you'll see here we have no processes shown. But what this is going to show us is as we are running different tools, it'll show us how long things are running for, if it's complete, so on and so forth. So this is a great tab to know about. And then moving on, we have our toolbox tab. This is where all of the different tools live that we will be using today. They're organized into these handy little folders that we can then expand using these little plus signs to grab the tools themselves. And then finally, if you have any favorite tools, you can go ahead and create a favorites folder here. And that will be a collection of all the things that you guys are using the most. So you can just have a quick access to it. Now up in the top right-hand corner, if you're watching this as a recording, it's probably underneath my video. There's going to be some buttons where you can have a look at things like designing workflows. Maybe you have some plugins that you'd like to use for CLC main workbench or even the Genomics Workbench, so on and so forth. But one thing in particular is going to be this support button right here. So if you ever get stuck or if you need an extra hand in figuring out how to do something or run into a technical issue, please feel free to reach out to our support folks. They are always ready and waiting for answering questions, and they typically get back to you rather quickly. So that's a great thing to know about. As well as that support button, we do also have this little help menu at the top of the screen that in this will also contain a whole wealth of information like online tutorials if you'd like to review maybe PDFs of what we're going through today. Another thing that we'll be visiting today is if you wanted to import some data to play around with while you're getting your feet wet with some of these tools, we have this import example data. And then, of course, we have that contact support functionality here as well. Okay, so now that you guys have an idea of what everything is on the screen, let's get right into it. So first things first, we are going to work on our alignment and construction of our phylogenetic tree. So the first thing that we want to do, of course, is import the sequences that we want to align. So that's going to start here with this import button. So we're going to go ahead and give that a click. When we do, you'll notice that we have our standard import and then our import metadata. So for right now, we're just going to be doing our standard import, which will then go out into a folder that I have saved a bunch of FASTA files into. So here we can navigate to that folder using either the Add Folders or Add Files button. In this case, I'm going to Add Files, click that button. And it will pop out to a spot in my laptop that I have saved some example data to. And I'm going to go ahead and choose that .fa example data here. So this is all of our sequences that we want to create an alignment with. So I'm going to go ahead and click on Open. And that will add it into the Selected Files and Folders screen. Once I'm good with that, I'm going to go ahead and click on Next. One thing that you'll notice is at the bottom here, we have these options. I'm going to leave it as an automatic import, but if you wanted to force a specific import type, you do have the ability to do that. So once again, we're going to go ahead and click on that Next button. And here we're just going to simply choose where we want to save it remember inside of our navigation area. So we see kind of a carbon copy of what our navigation area looks like. So for this case, let's go ahead and just set up a new folder that I'm going to call webinar. And click on OK, and I'm going to save that into our CLC data folder. If you wanted to maybe drop this into a higher level folder, feel free to grab and just drop it wherever you see fit here. So here we have our folder webinar, and once I am satisfied with that, I'll go ahead and click on Finish. And what this is going to do is it's going to now import those sequences into that webinar folder inside of our CLC main workbench environment. You can see how we're coming along with this process by visiting the left-hand bottom corner box here and flipping over to the Processes tab. And you'll see here that those sequences imported rather quickly. So we are good to go on to our next steps. So let's go ahead and have a look at our actual imported sequences here. So I'm going to open up our webinar folder using the little plus sign, and we'll see that we have our data here. So I'm going to give that a double click. Now you'll see that we have a whole bunch of different sequences. I believe it's about 80 or so that we're going to want to now bring into an alignment. You see here's all your base pairs, and of course you can mess around with how this looks by changing some of the settings over here on the right-hand side of the screen. Now the first step that we're going to do is we're going to visit our toolbox, give that a click. And inside of our toolbox, you're going to notice our collections of different smaller toolboxes, we'll call them. So the first thing that we're going to want to visit is going to be this Alignments and Trees toolbox drawer. We'll call it a toolbox drawer. That seems like a good analogy there. So when we open up this drawer of Alignments and Trees, you'll notice that we have the individual tools themselves. So the first one that we're going to pick from is going to be this Create Alignment. So I'm going to go ahead and just double click on this Create Alignment. And that will pop out a wizard-driven guide that'll show us exactly how to generate this alignment and what we need to grab to generate this alignment. So the first thing that it's going to ask is where to run this. So I'm just running it on my laptop, but if you guys have maybe a server or a cloud instance, you'd be able to choose to run it on one of those instances. But today I'm just running everything on my Hewlett-Packard laptop. Next thing we're going to do is going to go and grab those sequences that we just imported. So if you remember, we popped them into this webinar folder. So I'm going to go ahead and just highlight this phylogeny module example data, click on the little arrow over button, and you'll notice that it'll pop up in this selected elements pane over on the right-hand side. Once we are satisfied with that, we're going to go ahead and click on that next button. Now here is where we can set some parameters for our alignment. We can look at the gap cost. We can check and maybe go with a more accurate but slower alignment, so on and so forth. So one thing that you might notice as we're going through all of these wizard screens is that there's always going to be a help button in the corner of the pop-up. So if you're confused as to, hey, what is a gap cost setting? Why do I care about it? And do I want to change it? You can go ahead and just click on this little help button, and that will bring you to our help documentation that is specific for the view that we are clicking into right at this time. And so very, very helpful to give you a lay of the land of all of the different settings that we have available in the main workbench. Now for today, I'm actually just going to leave everything at default and let it do its thing on the fast list accurate alignment. And we're going to go ahead and click on that next button. Here we can choose to either just open the result or save it into a folder. That's exactly what we're going to be doing today, is saving it. You'll notice that one of the other things that you're able to do in the main workbench is create logs. So this is really, really helpful if you have, say, a public health lab or a clinical laboratory where you need to make sure to generate all sorts of documentation behind what you did with all of these samples and what they traveled through. So feel free to select this little checkbox if you wanted to create a log alongside generating your results. So here I'm going to choose Save, click on my Next button. And once again, just choose where I want to save this data into. So here, I'm just going to pop it right into our webinar folder that we've created and click on Finish. Remember, you can always organize inside of these folders as well with new folders if that's something that you'd like to do. But here, I'm just going to go ahead and click on Finish. I'm going to pop over to our friend, the Processes tab, to see exactly how long this is taking. Should be pretty quick. You can see we're already it's done before I could even finish my sentence here. So that fast alignment took all 80 plus of those sequences and created an alignment really, really quickly. And you can see that that result is popping up in our webinar folder up here. So you can see here's our alignment. So I'm going to go ahead and give that alignment a double click. What that's going to do is it's going to open up a tab of our alignment here that we can now go ahead and have a quick review of. So there's a lot of colors and things going on with this alignment. So one of the things that I definitely recommend is playing around with these settings that are over on the right-hand side of the screen to get things looking the way that you would prefer them looking, so you can have an easier time reviewing all of these different alignments. So maybe one of the things that we can change is the spacing. Perhaps you don't want any spacing between all of our nucleotides, so we can select to no spacing. The next thing that perhaps I'd want to do is change the way that these sequences are wrapped. So if I wanted to make sure that all of the sequences stay on one line per sequence, I can go ahead and choose this no wrap. If you wanted them to kind of wrap around the screen, so you don't have to scroll right, you can change that to that auto wrap or the fixed wrap. Another thing that can be pretty helpful is to change the matching residues into dots. So for me personally, it just makes things a little less cluttered on my screen so I can really hone in on where things are not lining up. So here you can see all of the matching residues now have been changed over to dots. Another thing, you might find these colors a little bit distracting and hard to see through. We can go ahead and change that color scheme over here. We can actually shut it off altogether by clicking perhaps on some of these checkboxes. So if I shut off the background color, all of the colors go away. If you wanted to just simply color the actual nucleotide identifiers here, feel free to change that to foreground color. So now we're able to go through and review our alignment to see where do we have a lot of different differences happening, so on and so forth. And you can scroll using this little toolbar at the bottom all across your sequence to see how things are lining up. Another thing to mention is going to be our zoom tools that are located at the bottom of the screen. So for instance, maybe you wanted to see everything fit into the screen. We can click on the zoom to fit middle icon here, and that'll snap all of our sequences to fit onto the screen. Of course, you'll lose your nucleotide level information. So if you wanted to go back to that base level, you can choose to zoom to the base level here. You can also use the magnifying glass to zoom into an area of interest, or the slider to increase or decrease how deep you're going into the sequence here. Now, if we wanted to say save this alignment to now go ahead and create a phylogeny tree, we can go ahead and do so by clicking on the Save button up at the top here. Another interesting thing that we can do is potentially export the sequences that we have down here. We can export the graphics themselves. So if you wanted to include a picture of this alignment, feel free to do so. Another thing that you can also do is right-click on the tab itself, and you will be opened up to a whole bunch of other menus for how to save or print or export some of these views here. So now that we have our alignment created, let's go back to the slides and just quickly go over exactly what we did here. So first things first, we went ahead and used that import button to grab our files and import them using this wizard here. So we went and looked for the files, grabbed them, and saved them into our CLC main workbench. We then went and chose the Create Alignment tool, which also opened up another wizard that'll walk us through the setup for the actual alignment itself, which at the end of the day will allow us to save and finish this, this alignment. Now, from there, what we did is we double-clicked on the actual alignment result itself. I showed you guys how to customize the view in the center here, as well as different menus to manipulate and change what you have in the center here. So let's go back to the software itself. Now, one thing that some folks might want to, might be interested in is changing what they have in these traces. So perhaps there's an issue with, say, this area right here in the sequence. Maybe we wanted to add something in or maybe move it over a little bit. We do have the ability to do so. So what I just did is I went ahead and using my cursor, I just highlighted the area that I would like to edit. And then from there, I'll just right click on this little purple box. And you can see here that we can add in perhaps an annotation for a troublesome area. Maybe we'd like to delete it or add in some gaps. Or maybe we just want to redo the alignment for this small section itself again. Lots of different options that we can do here. So again, what I did is I highlighted the area that I'd like to manipulate. Once it's this purple blue box, all I did was right click on that purple blue box. And here we have the menu to say, add some gaps before or after, or manipulate the sequence in various different ways here. And then, of course, once we've manipulated the sequence, we'll want to go ahead and save it if we did add in or change those sequences in any way, shape, or form. And that we can do using the Save button that I showed you guys up in the top left-hand corner of the screen. So now that we are done with this alignment, I'm going to go ahead and close these tabs just to keep our screen tidy. What we're going to do now is get into our toolbox and open up that alignment and tree drawer within our toolbox once again to go ahead and create a tree. So here I'm going to go ahead and double click on this Create Tree tool. And we're going to utilize that alignment that we just made together and generate a phylogenetic tree here. So first things first, it's going to ask you what alignment do you want to use to construct your tree. So here, let's go ahead and visit that webinar folder that we set up together and choose the alignment that we want to pop into our phylogenetic tree. So again, we're going to use our little arrow over buttons and make sure that it's in our selected elements pane right here. So next thing that we're going to do is click on our next button. And then from here, we have a bunch of different options that we can choose from, depending on how you want to construct your tree. Feel free to visit these different options here. And of course, if you have any questions about what any of these are, feel free to click on that Help button, and it'll give you a lot more detailed information on these options themselves. You can also perform some bootstrapping analysis, so feel free to leave that as default or change it to suit your mood. Another thing that you might notice is as I'm hovering over specific things in these options, you will also get a nice little tooltip just quickly explaining what these different options are if you don't want to get into that help menu right from the get-go. So once we are satisfied with our options, I'm going to go ahead and click on Next. For today, we're going to leave all of this stuff just as default here. So I'm going to go ahead and click on Next. And then, of course, we're going to save it. We're going to save it using this Next button into that folder that we've been creating today, that webinar folder. And I'll go ahead and click on that Finish button. This will quickly create a phylogenetic tree, and you can see it pop up rather quickly in our webinar folder here. But again, feel free to visit that Processes tab if you want to see what the status is for the tree construction. So let's go ahead and visit our webinar folder and double click on our alignment tree data here. So we're going to go ahead and double click on that, and it's going to open up our tree. Now from here, we have all sorts of ways that we can manipulate what we see in the middle here. So perhaps you are super happy with the phylogram layout, feel free to keep it. If you wanted to say change this over into a cladogram, feel free to do so. Or maybe you prefer a circular format for these different graphs. Feel free to change that to suit your mood here. Now down below, you'll also notice that there are settings for all sorts of nodes and labels and things like that. But you'll also see that we have this metadata section here. So one of the cool things for tracing back kind of lineages of the sequences and bugs that we've collected in these samples is maybe we want to see, are any of these kind of grouping based on some of these sample attributes here. So let's go ahead and add in some metadata that has those sample attributes included here. So what we're going to do is we're going to visit down in the bottom left-hand corner of our view screen. We're going to flip over to this table view. So show table. Once we've clicked on this little icon that looks like an Excel spreadsheet, what we're going to do is we're going to import some metadata. So I'm going to go ahead and click on that import metadata. And what this metadata is it's simply an Excel file that has all of our samples listed, as well as different attributes like what country was this sample collected in, what type of water was this sample collected in, so on and so forth. So imagine you can tack on as many metadata parameters or attributes as you see fit to color your dendrogram however you'd like here. So what we're going to do is going to go out and use this browse button at the top to go find my metadata file. And you'll see here I have an Excel spreadsheet that again is just a list of the sample identifiers and a whole bunch of different columns of other different metadata points here. So I'm going to go ahead and click on the OK button right here to grab that specific metadata file. And you'll notice that we have a preview down here. So this again is exactly what that Excel spreadsheet looks like. So here is our strain, here's the sequence of the strain, and then here's all of those juicy tidbits for where we collected these samples and maybe how we want to sort and color our dendrogram here. So once we're good with that, we're just going to go ahead and click on Finish. So now we have that metadata attached to our samples. So let's go back to our dendrogram view here. So I'm going to click on this little dendrogram icon at the bottom. And let's visit our metadata section now within our settings on the right-hand side. So I'm going to open up metadata, and I'm going to want to actually change all of these black node dots over to be colored by what type of water these were collected from. So what I'm going to do is I'm going to visit our node color, open that up, and I'm going to say, hey, I want this colored by water type. And you'll see that we get this key that shows up on the view itself. And now we have all of our different nodes colored based on where they were collected. And we can see that things are definitely seeming to sort themselves into the different water types here. So remember, all of these settings that are over here on the right-hand side will also carry over to anything that you might want to export using these buttons at the top here. So maybe you want to grab this radial phylogeny tree and pop it into a presentation somewhere. Using these colors by water or any of the other various selections that we have on the right here, feel free to do so. So with that, let's just review some of the steps we went through to create this tree. So back to our slides. So first things first, we went into our toolbox and we grabbed our Create Tree tool, which then launched our little wizard guided step-by-step, where we went and grabbed that alignment that we created together and put it into the selected elements pane, and then chose some different settings if we wanted to tweak how we're constructing the tree there. And then we went ahead and clicked on Next, and then finally save and finish. What we did was then manipulate what we saw in the center view here by looking at our tree layouts and maybe changing them from a phylogram over to this radial view or so on and so forth. But mostly we wanted to really add in our metadata so we can take advantage of that metadata and color code these sample dots. And we did so by visiting the view icon at the bottom of the screen there that looks like a little Excel spreadsheet, which then opened up a wizard asking, hey, where is this metadata stored that you want to drag into CLC? We had a nice little preview of it, and then we went ahead and clicked on Finish. And then from there, we can tweak those metadata parameters in the settings to be colored however we see fit, whether it's the shape or the symbol size or the color, things like that. So I hope I was able to give you guys a really nice overview of how to create an alignment in the CLC main workbench, as well as constructing these phylogenetic trees. So at this point, I would love to take a break to see if there are any questions that have come up while we've been chatting.
Thanks, Kristen. So thank you everyone for submitting your questions to the Q&A box. So we did have a couple of questions that came in. And so the first question is there a way to see which alignment I choose to create the tree from?
Absolutely. So one of the cool bits is CLC main workbench is really great at keeping track of everything, even if you aren't the greatest at keeping track of everything. So at the bottom of the screen here, you'll notice that we have a few different icons. One of them is going to be this show history, and that is going to show us exactly what we did and who did it and where we got the data from. So here I see originates from this particular alignment. So really great place if you've lost track of what you've been doing to get back to your kind of original files that you've used.
Awesome. So just to let everyone know, I just launched a speed poll just to make sure that we are, how do we feel about Kristen's speed? So you guys can just fill that out just to give her some feedback in that sense. So another question that came up is, what is the difference between the CLC main workbench and the genomics workbench? I know it was mentioned earlier today.
Yeah, great question. So think of it this way. Main workbench does not have any tools to handle NGS type data, so next generation sequencing data, whereas CLC Genomics Workbench does. So everything that you see here today in the CLC main workbench is also included in the CLC Genomics Workbench. So you can think of the Genomics Workbench as an upgrade from this main workbench to handle your NGS type outputs.
Awesome. So another question that came in is, how do you, or can you, rename the outputs in the phylogenetic tree?
Absolutely. So if you wanted to rename your outputs, say, in your CLC data folder files here, these the way they name them is just, kind of not very descriptive. All you have to do is just highlight the thing that you want to rename, right click, and then you can see that we have this rename functionality here, as well as a bunch of other cool things. So maybe you wanted to copy this and pop it into another folder. You have the ability to do that as well.
Okay. And so we had a question, can we add different sequence files and then do an alignment for all at once, or do we have to upload one file at a time?
Yeah, so where I showed you how to do that, create alignment, you can grab as many sequences as you want to align here. So for our case, all I did was just grab our one collection of sequences, but imagine you can grab numerous sequences that you've uploaded using your import button up at the top here. And just as long as you're dragging them all over into the selected elements, it'll run that alignment across all of those different sequences.
Awesome. And so just one last question. Can you import ABI file formats if you're starting from a sequencing file?
So here are all of the different file formats that we can import here. So let's look and see if the ABI files are included. I don't know, Araceli, do you know if we are able to do that?
That's a good question.
Or Sean? Sean's on the line. He is the guru in all things CLC. So we can get back to you with that answer in the chat box. Oh, Deb,
Yes.
to the rescue. Yes, the answer is yes. Great.
All right. Okay, so we still have a lot to cover, so we'll go ahead and continue, but keep typing in those questions in the Q&A box. We really appreciate that. And thank you for filling out the speed poll. It looks like your speed is just fine.
Awesome. Excellent. So before we move on, I just wanted to see from you guys, who thinks that utilizing that, the ability to color those different nodes by different colors and different pieces of metadata, is anybody here using that? Show hands. Are you guys going to be using that in the future for your experiments? Yeah, me too. I think it's a really awesome way to visualize your data and just get a quick kind of overview of, hey, what is similar, what is not, so on and so forth when you're looking for lineage type information. Awesome. So let's pop over into my slides once again. So we're going to move on to the next bit of the presentation here. So that is going to be our Stanger sequencing analysis. So here's where I'm going to show you how to import those sequences. You'll be able to trim them in a standalone trimmer, or you can run your assembly using these tools where trimming will take place inside of that assembly. So let's go ahead and kick back over into the software itself. I'm going to close this tab just to clean things up. And the first thing that we're going to do to allow me to show you guys how to do this is I'm going to go and grab some example data. And where we can find this is going to be in the help menu at the top of the screen here. And you'll notice the third thing down is the ability to import example data. So this is really handy data to get you guys used to using some of these tools here. So what I did is I just went ahead and grab that data, and you'll see here that it's going to import all of that data into a folder of my choosing. But luckily, I have already done this, so we don't have to wait for this timer to go down here. So let's go and visit that data. So at the very top of the screen here, you'll notice that I have this MWB webinar folder that I set up prior to this. And in that, we have our example data. And inside of that example data is a whole bunch of different data and sequences that we can play around with to learn the tools better. So first things first, let's go ahead and run a trim tool on some of these sequences that we have imported using this example data. And just a quick reminder, if you're importing your own sequences and not this example data, go ahead and click on that import button and use that wizard just like we did for our very first part of this webinar today. So what we're going to do is we're going to visit our toolbox at the bottom, and we're going to open up the toolbox drawer that is titled Sanger Sequencing Analysis. And you'll see there's a bunch of different tools in this toolbox drawer. So here, first things first, I'm going to show you how to trim your sequences if you want to do a standalone trimming here. So go ahead and give that trim sequences a double click. That'll open up a menu or a wizard that'll guide you through the whole rest of this process here. So first things first, we want to grab our nucleotide sequences that we have either imported or grabbed from our example data. So here you can see in our example data folder, we have this sequencing data folder. I'm going to open that guy up and see that we have a whole bunch of different reads that we can select and import into this trim sequences tool. So I have gone ahead and selected 9 different sequences to trim. Once I'm good with that, I'm going to go ahead and click on this next button. And here is where we're able to select some different parameters. For our case, we are going to leave everything at default. But if you wanted to tweak some of these parameters, feel free to do so. You can find more information about these parameters in the help menu. And you also do have the ability to perhaps trim out some vector sequence from your sequences if that's something that you have in your data here. But again, we're going to leave this stuff at default and move on with our next button, where we'll then be able to save these results and perhaps even create a log that will capture all of this trimming information here. So once we are good with that, we're going to just go ahead and click on that next button and save it into whatever folder we see fit. So I'm going to pop this into our webinar folder and click on that Finish button. So what this did is it went ahead and ran a standalone sequence trimmer. So I'll go ahead and open up those trimmed sequences that we popped into our webinar folder. You can see here's our 9 sequences, and it has trimmed alongside the name so we don't get things confused. And what I'm going to do is I'm just going to go ahead and double click on one of these sequences to show you here is the actual trace. And here are the annotations for these spots that the trimming tool decided to trim. So one thing to mention about this trim tool is it doesn't actually remove those sections of data from your traces, because sometimes even though they might be of poorer quality, they do contain information that we don't want to lose. So you can see here, we've just simply added in this annotation here. So feel free to run that standalone trim tool if that's something that you would like to do. Moving on, we're actually going to go ahead and run the Assemble Sequences to Reference tool with all of these example data traces that we imported together. So once again, going into our toolbox, what we're going to do is go ahead and open up the Sanger Sequencing Analysis drawer. And what we're going to do is we're going to either choose to assemble the sequences. So if you didn't have a reference to assemble to, you could just simply choose assemble sequences. But for today, we're actually going to assemble all of these sequences to a reference. So I'm going to go ahead and double click on Assemble Sequences to Reference, which launches another handy little wizard here. We're going to go and grab the sequences that we want to assemble here. So in this case, I'm going to go and grab those raw sequences again, so those sequencing reads that have not been trimmed. Pop them into our selected elements window. Once I'm satisfied with that, I'll go ahead and click on that next button. And here is where we can go ahead and choose what our reference is that we'd like to use. So I'm going to go ahead and open up this little folder right here to go and search through my CLC data folder for my reference sequence. So right here, we can see that we have this ATP8A1 mRNA reference sequence. So we're going to go ahead and use that as our reference. So I'm going to select it using those arrows over, pop it into our selected elements. Remember, you can import these ahead of time if you'd like to prior to running this actual tool. So I'm going to go ahead and click on OK. And then we do have some more options for tweaking these reference parameters as well. I'm going to leave those as default and click on the next button. Once again, we have some more alignment options, so we can change some of these options if you see fit. Another spot that you might want to take notice of is this trimming options. So if you created your trimmed sequences ahead of time, like we just did with that trim sequences standalone tool, you could go ahead and use the existing trim information from that, or you can leave it unchecked and go ahead with the trimming done inside the assemble sequences tools here. And again, always feel free to visit that help button if you want more information. So I'll go ahead and click on the next button and tell CLC where I want to keep all of these sequences, all this assembly. So here I'm going to go ahead and save it into our webinar folder. So I'll navigate to the bottom here, select our webinar folder and just pop it in there. Click on finish. And behind the scenes, we will have our assemble sequences to reference running behind the scenes here. So let's go back down to our webinar folder and open up our assembly that we just created together. So I'm going to go ahead and double click on the forward one assembly right here, which will open it up in that center pane here. If you guys remember from the last piece of our webinar today, we have all these zoom icons at the bottom that will allow us to zoom in or zoom out on these sequences that we've aligned here. So first things first, let's use this zoom to fit to bring everybody into the screen. So here's all 9 of our sequences that we've decided to align here, as well as a bunch of conflicts that seem to have arisen during our assembly. So you can use these buttons to zoom in or zoom out, whatever you see fit. So I'm going to stay here on our nucleotide level view here. You can scroll left and right to have a look at these sequences as well. So a couple things before we start plugging away with those conflicts is you can change how things are viewed at the center here. You can change the level of compactness that we see for these layouts. So maybe you don't want to see those traces. Feel free to choose the different levels of compactness. In this case, I'm just going to leave it as not compact. Again, with the wrapping, if you wanted to make sure that all sequences stay on the same line, we're going to go ahead and choose that no wrapping. And then, of course, if you wanted to change maybe some of the residue coloring or nucleotide info, things like that, you can absolutely do so as well. So typically the next thing that we want to do when we've created an assembly is go through and review all of the conflicts that might have arisen between our traces that we've imported where they differ from our reference sequence. So in the center of the screen here, you can see that we have this consensus sequence. So this is taking into account all of the trace information that we've imported, matching it to the reference and saying, hey, this matches, or hey, it doesn't match. So what we're going to do is we're going to actually use this find conflict button to quickly just go through all of the different individual conflicts that arose and resolve each of them. So what we're going to do is go ahead and click on this find conflict button. What that's going to do is it's going to bring us to our very first conflict that we see between our traces and our reference sequence. So you can see in the reference, we're supposed to have a C. This first forward one trace looks like a very clear-cut C. And then down here, we have some poor quality sequence. However, in reviewing this, it doesn't actually look to me like there is a gap here. It actually looks like there was just a low quality C that was actually detected at this point right here. So what we're going to do is we are going to resolve this conflict by going ahead, and we are going to highlight either in the reference right here, so our conflict, we can highlight that nucleotide, so it turns this like bluish purple, right click, and we could choose to transfer this C to all of the reads that we have down below. So this will resolve this conflict and change our actual trace. And this is exactly why we're getting this warning saying, hey, you're changing your trace. Are you okay with that? You can choose to shut this dialog off if you'd like, or you can just simply click on the OK button. So what this is going to do, it's going to go ahead and change this C over to the C. And it looks like I actually highlighted the wrong C here. So let me just highlight the actual conflict C. Again, right click, transfer selection to all reads, click OK. And now you can see that we now have changed into this resolved conflict status. So using this find conflict button, you can see how you can just quickly zip across your sequence, move to the next conflict, resolve it, and move on with your day to ensure that your consensus sequence is matching with your reference sequence here. So once again, we have our C that is showing a conflict here. Looks like this first trace, we do also have a C. And then we have some messy sequence quality, but again, I believe, there's also a C at this point as well. We can see that peak behind that messiness. So I'm going to once again go right click, transfer selection to all reads, go ahead and click on that OK button. Now we have this resolved conflict. So once we've gone through and resolved all of these conflicts, we can go ahead and save this assembly using our save button. We can also choose to export it or export a graphic here as well. So just with a show of hands, who here thinks that using that find conflict button is really going to be super helpful in cruising through your sequence, resolving all of those conflicts in a very orderly fashion? Yeah, I think it's a really great way to kind of review your whole sequence as well. Wonderful. So let's just go back to our slides and review exactly what we've done so far. So we're in the Sanger sequencing analysis portion of our webinar, where we went ahead and we imported reads, or we grabbed the reads from that example information that we have in the help menu. So you guys will be importing your reads. You'll be using your standalone trim sequences tool. If you'd like to trim the sequences, you will select those particular sequences you'd like to trim and set the parameters and save and finish those trimmings. If you would like to just have a look at those sections that got trimmed, you can see here that an annotation is added into your sequence here, and that information is not removed from your data, just in case there's important stuff that lays in that trimming area. And then what we did is I went ahead and showed you guys how to assemble those Sanger sequences to a reference. This is going to be pretty much one and the same, whether you're doing it without a reference or with a reference. You can choose your tool from the toolbox here. You're going to go ahead and choose those different Sanger sequences you want to assemble. You're going to choose your reference genome that you're choosing to assemble to. And then, of course, some parameters here, which then we went ahead and we saved and finished. We then cruised through and I showed you how to optimize that view in the center to suit your mood. I showed you how to zoom in and zoom out to different levels of your sequence here. And then we spoke about how to resolve some of the conflicts that you see popping up between all of your traces and your consensus sequence as well as your reference sequence. So here we can use that find conflict button to cruise through your sequence and resolve each of those individual conflicts that we might see arising in our sequence data here. And then from there, you can go ahead and save those sequences once they have been all tidied up by having a look at all those conflicts and move on with your experiment. So the next thing that we're going to be getting into is going to be our cloning portion of the talk here. So during our cloning portion of the talk, what we're going to do is we are going to use some restriction-based cloning to insert this particular gene of interest into a vector. And how to do this, we're going to go ahead and we are going to, first of all, create primers to amplify our target region of interest. We're then going to go and choose what restriction enzymes and sites we want to use to snip into our vector and put that insert in there. We're going to tack those restriction enzyme sites onto our primers so we have some way of getting our insert into the vector there. And then we'll go ahead and just run an in silico cloning experiment to make sure that everybody is lining up the way that it should. So first thing first, what we're going to do here is we are going to go ahead and create some primers. So I'm going to go back into the software itself. And we are going to start making some primers here. So I'm going to close our assembly that we've been working on together. And what I'm going to do is I'm going to go up into our MWB webinar folder that I have gone ahead and downloaded the example data for to. And I'm going to open up this primers folder. So here we're going to have our sequence for our insert that we would like to stick into our vector of interest. So I'm going to first and foremost double click this insert sequence here to have it pop up in the center here. Now remember, we can change how things are looking using all of these different sequence settings on the right. In this case, I'm going to just make it easy on us and use some of the zoom tools that are at the bottom right-hand corner. What I'm going to do is I'm going to take and snap this sequence to fit the window for the entire insert itself. So I'm going to go ahead and click this center button. And that's going to take the entire sequence and pop it into this screen, all fitting into one spot. So now we can see here is our insert. If you wanted to shut these flags off, if they're overlaying on top of important information, you absolutely can do so using our sequence settings to maybe shut off some of these restriction enzyme flags to visualize exactly what all these sequences are behind the scenes. But trust me, there are the actual insert sequences that are in this yellow and the blue, as well as all of the important bits that come with this insert itself. Now, what we want to do is we want to stick a forward primer right upstream of the insert itself. So we really want to pop this primer into that CMV promoter area right here at the beginning of our gene of interest that we're going to be inserting into the vector here. So what we can do is we can just very simply on this black line that shows up below these annotation arrows, we can highlight the section that we would like to stick this primer in. So for instance, let's go ahead and just select this area on this line to pop the forward primer in. So you'll see that the selection turns the bluish purple. Once we've selected this area, I'm going to go ahead and right click. And here, what we can do is go ahead and tell CLC Workbench exactly where we want to pop this primer. So in order to do so, we're going to go ahead and switch over to our primer designer view. So right now we're just simply looking at the sequence itself. So we're going to go over and open up this primer designer, which once again is going to look very much like what we were just looking at, except we're going to have these lines that are generated below. So on this screen, what we're going to do is we're going to go ahead and select the area that we would like to stick that forward primer in. We're going to right click on the blue purple area. And the very first option here is going to be forward primer region here. What this is going to do is it's going to tell CLC, hey, we want the primer to sit somewhere in this area, the forward primer. And then using the zoom tools at the bottom here, I'm going to go to this I'm going to go to the selection tool right here and just zip right into our selected region, where now you can see all of these dots showing up on all of these 5 lines that are populated on the primer design screen. So first things first, each one of these individual lines is specifying different length primers. So if you want a length of 18, here's your 18 line, 19, 20, so on and so forth. And then each of these individual dots is going to show where the start of a primer could potentially be. Now, depending on the settings that we have listed over here on the right-hand side, these dots will either be red or green. So if we are not meeting the criteria or the parameters that we've set out on the right-hand side, the dots will be red. If we are meeting the criteria, the dots will be green. So you can see here that we have lots of spots that we can start our primer, our forward primer on based on all of these different green dots. So I'm going to go back and zoom back out to see our entire sequence on one screen. So now we have our forward region specified, and that's exactly what this little arrow is. Now we can go and select where do we want to pop the reverse primer. Remember, we're doing Sanger sequencing, so you want to keep those fragments a little bit more on the reasonable side of things when performing PCR. So what we're going to do is we're going to choose to put the reverse primer right at the beginning of our insert of interest here, and highlight that area on this black sequence right here. Right click and click on this reverse primer region here. So this will now pop that reverse primer into that area of interest. And then once we are satisfied with that, we have now set up our forward region and our reverse region. So we can go ahead and calculate what our different primer pairs would look like. So here we can go ahead and click on that calculate button. We're going to have a look at our primer parameters that we have chosen. We can go ahead and change some of the parameters for our combinations if we would like to. But at the end of the day, we're just going to go ahead and click on calculate and leave everything at default. So here what you can see is after we've clicked on that calculate button, what CLC is doing is it's mapping out different primer pairs that exist in those intended regions of interest on our insert, and show us a whole list of different primer pairs that we could potentially use down below. So what we can do is, perhaps we like this very first primer pair. We can go ahead and click on this primer pair itself, and then right click. We can mark where these primers sit on our sequence by using this mark annotation. Or we could just simply save these primers as what we want to use for our cloning experiment. So I'm going to go ahead and save these primers into our webinar folder and click OK. And then if we scroll down to our webinar folder, you'll see we have our reverse and our forward primer sitting here ready for some manipulation here. So far, we created some primers to amplify our insert of interest So to do so, what we did is we just double-clicked our sequence of interest. We popped over into our primer designer view, and then just simply told CLC where we want the forward and reverse primers to sit. Once we were good with that, what we did is we went ahead and clicked on that calculate button. Maybe we changed some parameters and then hit that calculate button once more. And then from here, what we can do is see all of the different primer pairs that we could potentially use, and then save those primer pairs into a folder for further manipulation here. So we have gone through a ton of information so far. I would love to take another live Q&A break to see, are there any other questions that have popped up in chat, and check in with you guys to see how we're doing.
Awesome, Kristen. So before we get into the Q&A, I'm just going to go ahead and launch today's last poll, just to get an idea, of how useful you found today's webinar to be so far. So in terms of questions, if you guys have questions, please go ahead and utilize the Q&A box as you guys have been doing to type in those questions so we can go ahead and make sure that we're answering them. So a question that came up is, are there walkthroughs available for some of these tools?
Yeah, absolutely. So if you prefer learning using, say, a PDF, we do have lots of resources to do so. So I'm going to go back into the main workbench itself. You can visit this help menu at the very top and then go to our online tutorials. There you'll see a whole ton of PDFs that have very detailed walkthroughs on all of these different things that we've been going through today. At the beginning of my slide deck that you guys should have, I actually linked to all these tutorials as well. And the other handy bit about this is it will point you to where to grab example data from. So you can practice without having to mess around with your own data here as well.
Awesome. So I've go ahead and reposted the link to the slides that Kristen made in the chat box so you guys can have access to them. So a question that came up is, do we also have tools for CRISPR-based cloning?
So I believe the CRISPR tools are going to be in the Genomics Workbench, if that's something that you would like to visit. I don't know if Araceli, Dev, or Sean, if you have any more tidbits to add to that.
Yeah, so from my experience, users that are working with CRISPR typically utilize the tools that are found with the upgraded version of the main workbench, which is the Genomics Workbench. That one is awesome at handling NGS data, and it does provide you with some awesome tools to take a look at that CRISPR data. So if you guys are interested, we do have a trial link in the slides that Kristen provided, or you can also reach out to us and we'd be happy to help you as well. And I guess similar to that topic, do we have tools for 16S and metagenomic data?
Yes, so all of the tools that I am showing you today in the main workbench also exist over in the Genomics Workbench. And there are tons of different tools from metagenomics over there, 16S sequencing, all sorts of things. So all of your NGS kind of needs will be fulfilled through that Genomics Workbench. So again, all the stuff that we're seeing today is on the main workbench. And if you wanted to upgrade to that Genomics Workbench, all this stuff will also show up in that Genomics Workbench, as well as all the NGS stuff and metagenomic and all of that.
Awesome. So I'm going to go ahead and just, for those of you that are interested in taking a look at are working with microbials, and it seems to be a good topic, I'm going to go ahead and type in just like a link to our page. You guys can get more information on that. And so I'm just going to go ahead and do that in the chat box. And so another question that came up is, I noticed that you were using some of the tools and it has different parameters. How do I know what some of those parameters mean? Is there a way that I can find that answer?
Yeah, absolutely. So for instance, let's go and open up, say, an assemble sequences. And if you come across any parameters that you are confused about, please feel free to utilize this help button. There is a wealth of nitty-gritty information about any and all of those parameters that you can tweak and mess around with in all of these tools. So again, that help button is going to be your best friend when it comes to stuff like that.
Awesome. Okay, so it looks like we don't have any questions, but again, if you guys have any questions, please continue to use the Q&A box to type in those questions, and we will answer them as they go along.
Okay, sounds great. So we do have quite a bit of ground to cover today. So we may be spilling over from our 2:30 mark, so I apologize for that. But if you have to leave, if you have a meeting right up against this, feel free to leave. And we are recording this session, so you're not going to miss anything if you wanted to review that other information that we might spill over into. So let's get right into it to not waste any more time. So the next thing that we're going to be doing, so we've created our primers for our cloning experiment. The next thing that we're going to do is we want to add in some restriction enzyme sites onto the ends of our primer, so we're able to stick that insert into our vector. So first things first, we actually want to go and figure out what restriction enzymes we want to even use. So let's go ahead and in our example data folder, I'm going to go into cloning here. And in the cloning folder, we're going to see that we have both our insert of interest. I'm going to double click and open that. And then we have our vector of interest here. So I'm going to open up that vector of interest as well. Now you'll see that we have these two tabs. If you wanted to split screen to make things a little bit easier to look at, feel free to right click on any of these tabs, click on view, and then you can split horizontally or vertically. So let's split these guys horizontally. So at the top, we have our insert, and then down the bottom, we have our vector. So let's put it into a circular format just to have a little bit better view of the entire sequence itself. So in our vector sequence, I just popped it into our circular view using our tool at the bottom left-hand side of the view pane here. And now we can go ahead and have a look at our multi-cloning site to figure out which of these enzymes would be the best to use for our experiment of interest. In this case, what we're going to do is we're going to grab Handy3 and Zo1 as our restriction enzymes to get our insert into our vector. So now that we have chosen which ones we want to use, I'm going to go ahead into our primers folder where we've gone ahead and saved our forward and our reverse primers here. Actually, these are the ones I've already manipulated. So let's close these out. And actually, let's open up the raw primer sequences that I imported into our webinar folder. So here we have our forward primer sequence here. And what we're going to do is we are going to add in a restriction enzyme site as well as a few nucleotides of buffer to ensure that we have enzyme efficiency here. So first things first, we're going to go ahead and actually click on the, double click on the name of the sequence itself, which then in turn highlights this entire primer sequence. Once we've done that, what we're going to do is we are going to right click into our purple blue area and choose to either insert a restriction site after the selection, or we can choose to insert before the selection. So in this case, we're going to choose to insert it before the selection. So again, right click, insert before selection. We can go and grab our Handy3, which was the first enzyme that showed up at that multi-cloning site where we wanted to stick our insert. So I grabbed the sequence or the restriction cut site for Handy3. It's going to add that onto the front of our primer here. And then, of course, we want to add a couple additional bases just to make the enzyme happy, right? We want some kind of runway before those cut sites for enzyme efficiency. So once I'm satisfied with that, what I'm going to do is I'm going to go ahead and click on OK. Now you can see here that we have our Hindi 3 site as well as our little buffer zone for our enzyme. Once we're good with that, you're going to go ahead and click on the Save button and save that primer sequence with the new things that we just added onto it. Now for the reverse primer, we're going to do the same exact thing again. So I'm going to double click on the reverse primer. It's going to open it up. I'm going to double click on the name. It's going to highlight that whole sequence. I'm going to right click on the whole sequence. And in this case, this primer has actually been designed to match the negative strand. So we are also going to include a restriction site before this primer as well. So I'm going to insert the site before the selection. And in this time, I'm going to go ahead and choose our ZO1 restriction enzyme here. And again, add a nice little buffer prior to that cut site and click on OK. And now we can see our ZO1 cut site is included before our primer as well as that buffer. So feel free to go ahead and save those primers. Now, the next thing that we can do is run an in silico PCR to create the fragment that we are actually going to be physically inserting into our vector of interest. So what we're going to do is we are going to go down to our toolbox here. I'm going to close out all of these tools here, and we are going to go into our different toolboxes here. So one toolbox that we're going to be using is going to be our cloning toolbox, and you can see we have our restriction-based cloning here. But another thing that we're going to do is we are going to go ahead and have a look at all of these other different, toolboxes to try to go ahead and find the next tool that we are going to be using. So in primers and probes, you'll notice that we have this find binding sites and create fragments. This is our in silico PCR tool. So once again, once we opened up primers and probes, double click on find binding sites and create fragments. And at this point, what we're going to do is we are going to go ahead and choose the mRNA that we would like to amplify. So I'm going to go up into our molecular workbench webinar folder, and in our different cloning folders here, in our example data folder, you'll see that we have this mRNA sequence. So this is the mRNA sequence that we want to actually amplify. So I'm going to go ahead and grab that as our selected element and then click on Next. Here, now we're going to go and grab our primers that we're choosing to amplify this mRNA with. So again, we can go and find those primers of interest that we created. In this case, I'm going to go into our primers folder and choose the primers that we have created here with those cut sites as well as the buffer for the enzyme here. So here I'm going to grab those two primers and then just very simply click on OK. You can choose to tweak some of these parameters here, but once again, we are going to set at default and then click on the Help button if we have any more questions about that. But here we're just going to click on Next. At this point, we can choose to either open the results that we get from this in silico PCR, save this particular PCR product, or whatever we see fit here. So in this case, what I'm actually going to do is go ahead and just open this result rather than save it, because I don't need to actually save the results itself. All I want to do is actually save the fragment that is resulting from my actual PCR results. So I'm going to go ahead and choose this open for the result handling and then click on Finish. Here, what that's going to do is it's going to open our results into a separate window where we can go ahead and, let me close out of some of these, where we can go ahead and choose to save the resulting PCR product that we just generated here. So I'm going to go ahead and open up the fragment that we've saved prior. And you can save that fragment by using our save icon up here, or even just right-clicking on the fragment itself and choosing Save Fragment. So now that we have gone ahead and created our fragment of interest, we're going to go ahead and simulate a restriction-based enzyme cloning reaction here. So what we're going to do is we're going to open up in our toolbox our restriction enzyme digest cloning tool. So in the toolbox, we're going to open up the toolbox drawer for cloning that I showed you guys before. So in cloning, we have our restriction-based cloning tool. Kind of give that one a double click, and it will guide you through exactly what you need to do to create your construct here. So what we're going to do is we're going to grab that fragment that we made, as well as the vector of interest that we want to put that fragment into. So I'm going to go ahead and choose both of those sequences here, both the fragment as well as the vector, and click on Next. Here I'm going to go ahead and click on Save and pop that into our webinar folder for safekeeping. We're going to now go into our webinar folder and open up that cloning experiment that we just created together. So I'm going to close this out to clean the screen up a little bit, open up cloning experiment. Here we have our cloning experiment. So the first thing that we want to do is change over from just this very simple sequence viewer over into our cloning editor here. So it looks like a little vector or a horseshoe, whatever you want to call it. It's this little icon down the bottom here. So I'm going to click on that to pop open our cloning editor. And what I'm going to do is, first things first, I want to tell CLC which one of these two sequences are actually our vector. So right now we are selected on our fragment. So I'm going to go ahead and choose our vector here by selecting the dropdown, choose this TO vector. You'll notice that the illustration changes to our vector itself. And then what I'm going to do is just tell CLC, hey, this is actually our vector. So I'm going to say change to current. Now, once I have set this up as our vector, we want to tell CLC what are the restriction enzyme sites, cut sites that we're going to be using for our experiment here. So if you remember, it's going to be Hindi and Zo1. So what I'm going to do is I'm going to click on the Hindi annotation here. I'm going to hold down on my control key on my keyboard and also click on Zo1. When I do this, both of those restriction enzyme sites will be highlighted, and you'll notice that we have this selection down below. And what CLC is asking is which of these fragments are going to be our target vector? Is it going to be the little bitty piece that exists between those cut sites? Or is it going to be this huge 5,000 base pair section that exists on the outside of those cut sites? So here I'm just going to make sure that I'm selected on the correct vector sequence and go on with my experiment here. So now that we've set up our vector, let's flip back to our target that we'd like to insert into this vector by changing our drop-down screen here. So we're going to do the same exact thing that we just did with the vector, but to our insert. So what we're going to do is we're going to go and highlight our Hindi 3 restriction site, hold down on our control button, and choose Zo1. So here you'll notice that we'll get a few different selections asking us, hey, what is the actual insert that you want to put into your vector? Is it the tiny bit of sequence that's before Hindi 3? Is it the section that's between Hindi 3 and Zo1? Or is it that little tiny piece that's after Zo1? So of course, our insert is going to be this middle one, or this 3,000 base pair fragment here. So once we are good to go with that, and we've chosen everything that we are interested in choosing, we're going to actually go ahead and just click on this clone button, and this will run our in silico cloning experiment. When we click on this button down below, This is going to open up this menu to allow us to adapt the overhangs. So if you wanted to do a reverse complement if needed, feel free to do so using this little button right here. And this will just double check to make sure that your vector overhangs are matching up with your insert overhangs. And then once you're good with that, you're just going to go ahead and click on Finish. Now you'll have an overview of your construct where you can go ahead and inspect to see, what this looks like. So here is our insert of interest. This ATP8A1 gene has been inserted into our vector that is 8,460 base pair long. So I just want to thank you guys for sticking with us today. We went through a lot of information. So let's just have a quick recap of exactly what we went through in this last little bit of the webinar. So of course, we wanted to go ahead and try to figure out what restriction enzymes we wanted to use to construct our construct of interest here. So we went ahead and opened up those sequences and picked out the most appropriate cut sites. We then went and added those cut sites to the primers to ensure that we were able to stick our insert into the vector itself. So we double-clicked open our, we double-clicked open our primer of interest. We selected it. We chose to insert a restriction site for our enzyme of interest. And then, of course, we added that additional buffer to make sure that the enzyme was happy when it went to go cut the insert itself. We then ran an in silico PCR to generate a fragment, the actual fragment itself that we would be sticking into the vector of interest. So we did so by going into primers and probes. We selected the find binding sites and create fragments. We looked for the mRNA sequence for our intended sequence of interest. We amplified it with our primers that we created with our cut sites. And we then saved the fragment into our data here. Once we saved that fragment, it was available to then start off actual in silico cloning reaction here, where we went ahead and chose not only our vector of interest, but that insert that we created together. We chose our cut sites, and we told the software exactly, which of these fragments we were creating with our cutting were our vector as well as our fragment. And then we went ahead and created our construct of interest after going through a few more steps here that are outlined in those slides that we shared here. So with that, I thank you guys so much for sticking with us. And would love to pause to see if there are any other additional questions that came up in the Q&A box.
Thank you, Kristen, for this wonderful presentation. And thank you, everyone, for attending today and typing in those questions. So if you guys have any questions, please go ahead and utilize the Q&A box located on your screen to type in those questions that we can be able to answer them live. So one of the questions that came up is, how can I export the image of my construct and utilize it for publication? Is it an option in CLC?
Yeah, totally. So first of all, if you wanted to change some of the settings to make this pretty for a publication, feel free to do so. But if you wanted to actually export the image itself, what you're going to do is you're going to go ahead and click on this graphics icon here. This graphics icon will let you either just do the visible area or the whole area, and you're able to export it in a various amount of different resolutions and different file types to include in your publications or your presentations.
Awesome. And so just so that we can help the attendees today, can you highlight again how they can have access to tutorials for those of us that are newer to the main workbench and want to play around and explore? We don't have the data to do so.
Absolutely. So if you wanted to explore some of these tools and just test them out, what you can do is you can go into this help menu at the top of the screen here. And then in this screen, you'll notice that we have these online tutorials. So for instance, if you wanted to do your own cloning experiment, go ahead and click on online tutorials. That'll bring up a web page where you can go and grab the molecular cloning or the molecular biology tools tutorial that has this cloning in it, as well as links to practice data. So you don't have to wait around to generate data to try out some of these tools themselves. And then, of course, if you're under license for CLC, please definitely reach out to either support or out to your sales rep to maybe set up with some field application scientists to have a one-on-one type session as well.
Awesome. So we're going to go ahead and stay on for a minute or so just to make sure that we capture anyone trying to type in their last minute questions in the Q&A box. But again, thank you everyone for attending today and taking part in the polls. It really helps us, sort of get a gauge of how we're doing and how we can, do things better in future webinars. So again, if you guys have any questions, please go ahead and use that Q&A box located at the bottom of your screen. And so we'll stay on for like 30 more seconds or so just to make sure that we don't miss anyone's questions. All right, so it looks like there's no more questions. So I'm going to go ahead and just stop the recording.